My talk will be about the mirror neurons. And what I want to show you that now we change from the idea that is a specific set of neurons that have some property I explain in a moment, that the whole brain has this mechanism in the sense that the whole brain, or part of it, large part of it, work as a mirror system. But let me start from the very beginning. About 20 years ago, we discovered in the premotor cortex of macaque monkey a set of neurons which fired both when the monkey performed a certain action and when the monkey observed the same action made by the experimenter. In other words, there was a transformation from sensory information that the monkey received to motor information inside her brain. So you see, you transform something which is outside in the world in something which is in your brain. Before speaking in more detail about the mirror neuron, I must say that we discovered them when we studied the motor cortex. And that's very important because still the mirror neurons are connected very closely to the motor system. So what we did, we recorded single neurons using exactly those electrodes which has been mentioned by Edward before, the Hubel tungsten electrode. As a matter of fact, I was very proud because mine was very good. So I was doing by myself the tungsten microelectrode, and we recorded single neurons from motor cortex. What was new in our experiment that we used an ethological approach. Usually in the motor system, the people record the dynamic, the kinematics of movement. We recorded the behavior of the monkey during interaction with the experimenter. And the area which we studied most is the ventral premotor cortex, and more specifically area F5, which is here. Well, there was some surprises. The first surprise was that many neurons in this area did not code movements, but what we call it motor acts. If you stimulate, for example, the motor cortex, you see these twitches or movement, displacement of joints. But what we found that these neurons fire when the movements are organized in such a way to reach a goal. So we introduced the concept of goal in the motor cortex. And that was very important then to interpret other data. For example, in this neuron that you see here, the monkey is grasping with the mouth, it's grasping with the right hand, with the left hand. It's the same neuron. So the movements are completely different, but the goal is the same. At this point, somebody objected that in this case, it's a kind of synergy. Maybe mouth and arm are used in the same way. So we have been challenged to do this experiment. In this case, you have a normal pliers in A, and in B, a pliers, which is very strange, because in order to grasp, you have to open the hand. This has been a suggestion from a French postdoc. And if you remember, in France, the escargot, the snails, are eating using this strange instrument in which you have to open the head. It's very complicated, at least for me. But it was also complicated for the monkey. But anyway, the monkey learned it. So we have an experiment in which the monkey grasped the object using the normal pliers and using these reverse pliers. Now I will show you a monkey. And what you have to do is to correlate the action potential that already you saw in the previous lecture. They are sent to loudspeaker, so it will be tack, 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 and correlate what the monkey is doing and the discharge of neurons. Here is a grasping neuron. Here is using the strange pliers, and you have the discharge. Look here, the monkey is moving, nothing happens, but when the monkey grasps it, there is a discharge. So we put a potentiometer to measure the things, and you can see the potentiometer when it's going down the curve here. It means that the hand was closing, and here the hand was opening. So in spite of opposite movement, you have the discharge which correlate with the goal rather than with the action. Well, the other surprise was that many neurons in the motor cortex was responsive to visual stimuli. At the beginning, still with Gentilucci many years ago, we found that there were neurons. Imagine you have a neuron which fire when the monkey grasps it this way. If you show to the monkey a small object like a seed, there is a discharge. The monkey was completely still. So there was a transformation of this object into action. And now, more recently, in my lab, they found exactly the parameters which are necessary for getting this result. But anyway, the point is there were neurons which fired when the object was congruent with the action. The surprise was that, in addition, there were neurons which fired not when the object was presented, but when the monkey saw an action. Here you see one old picture. In B is the monkey grasping this small bar action potential. You see the discharge. And in A is the experimenter doing the same thing. And you have the discharge. The same is true for mouth. Here is another experiment. And the monkey grasped with the mouth. There was a good discharge. When the monkey sucked from a syringe, was nothing, although visually it's very similar. And the same is true for experimenter, grasping food with the mouth, good discharge, experimenter sucking from a syringe. No. And food presentation per se was not sufficient, so was not simply interesting or something salient for the monkey. Now I show you a film which is rather old, but it's still the best representation I have. Remember, you have to correlate the tac, 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 the action potential, with the behavior of the monkey. Again, a grasping neuron.
Fanno un altro così. Come prima, tiri. Ancora.
Same neuron, but now it's the person grasping.
Fai ancora. Fai.
You see, it's highly repetitive.
Provo questo.
That's the reason, you know why.
Prova con uno dei tuoi stimoli che lei conosce.
So it's grasping. It doesn't matter if grasping is made by hand or by mouth. And you see how it's, you can repeat any time you want. It's not something which is rare that you must do a histogram. When you have a mirror neurons, you can test it until it's alive. Well, what is strange here? That we have a visual action and then there is a discharge in the motor cortex. Here, the best explanation has been done by Marc Jeanneret, which Paul mentioned it before. He was really the first who understood the importance of motor system for cognition before our discoveries. And he wrote, a mere visual perception without involvement of the motor system would only provide a description of the visible aspect of the movements, but it would not give precise information about the intrinsic components of the observed action, which are critical for understanding what is action about, what is its goal, and how to reproduce it. How to reproduce it means imitation, but I am not going to talk about that. But the other, I think, is very important. So you understand the action of the other if you have it in your motor repertoire. Now I will show later some example. However, that's very nice, but one can say, well, what is the evidence that mirror neurons somehow are related to action understanding? And one thing that people object to say, what about other modalities? Why you don't try to use auditory modalities. If this neuron is related to understanding the action, it should be the same. Imagine that you're in your hotel and you listen to somebody walking, you understand very well the action, you don't need to see. And the blind people are very good in that, for example. Well, so we did an experiment. That I will show you our protocol, let's say. So the small bar are actions, potentials, and the histogram. You see that one experimenter tears a sheet of papers in two pieces and the monkey sees it, there is a discharge. Now the experiment consisted in placing a barrier so the monkey cannot see the action, but can hear the specific noise that when you break a sheet of paper, you can hear. And here is the answer. The response is still there, so it doesn't matter if the monkey sees or hears the action, there is still a discharge. And here is control with white noise. Personally, I like very much single neuron and hear them, but if you want to publish in an important journal, you must do some statistics and complicated things. And that paper has appeared in Science, so we have to do the population analysis and many tricks that you probably know already. But the important thing is S. S means only sound. We have 39 neurons, and blue is the sound which is specific for the action, and in red are the responses if you presented some other type of noise, of other action or white noise. Okay. The second evidence in favor of understanding is the following. Now the monkey will not see at all the object, but can guess that the object is there. So imagine that you are sitting, like monkey, and sees this video. This is a typical mirror neurons. The girl is grasping an orange, and you have the discharge. Oops. Here there is no object. The action is the same, but there is no object. Now we will close this the vision of the orange. You see, there is a discharge. The monkey can guess what will be. All of you can understand. If there is an orange and somebody is moving their arm towards do it, it's because you want to grasp it. And if there is no object, there is no discharge. Well, in other words, if you have sufficient element to understand an action, the mirror neurons fire. The point is that sometimes people criticize that, say, well, mirror neurons are a relatively small amount of neurons, how they can do all this stuff. Actually, the same effect is present in many other parts of the mirror, of the motor system. And the best example has been achieved by Roger Lemon and his group, Kraskov, in London. As you know, Roger is probably the best scientist in the field of corticospinal tract, and he recorded single neuron from corticospinal tract and found that many of them have mirror properties. That's his data. The red light is neurons which fire when the monkey sees the object, facilitation as we call it, and he has found something interesting. There is also a set of neurons which fire when the monkey grasps, but are inhibited when the monkey observes. So what we discovered, it's not only a group of neurons, but a system which discharge when you see an object. So you have, in a sense, the motor schema. When the motor schema that's inside yourself is excited because you see another people doing it, you understand the action. You have a kind of phenomenological capacity to understand the action. You don't need to think about there is a hand which is close to the object and now it's close, blah, blah, blah. So you understand it directly. Of course, another problem is how these neurons are generated. I don't know how they are generated, but I know the circuit. And that's we did together with Guillermin and Nelson. The experiment has been done in Leuven because they were very clever. You know that in order to do a fMRI experiment, the person should be still, cannot move, otherwise you have artifact. And the people said, with the monkey, it will be impossible. Instead, if you train the monkey to sit in a sphinx position and to observe a dot, when the dot changes the color, the monkey is reinforced. Well, at this point, the monkey can sit for 40, 45 minutes, ready to receive reward. So they trained the monkey first in a box, and when the monkey is ready, they place it in the standard scanner. So it was a cheap experiment. You don't need a special scanner and so on. So we did this experiment, and the guy, Nelson, was the guy who did most experiment, and then the other people also helped with anatomy. But the first experiment we did was just pure fMRI, look at 3 Tesla, which is here. And so when you observe an action, you have an activation in STS. This area is called superior temporal sulcus. There are many areas there, as a matter of fact. It has been studied by David Parrott in St. Andrews, and he described many interesting properties in this area. So this area is the first visual area, which became very complicated. Then it's parietal lobe, and now we have data that in the inferior parietal lobe there are many mirror neurons, and there is a transformation from action observation to action, real action. And finally, you see here is the frontal lobe, it's our area. I mentioned it before, all these people, because after this study, we did one in which we put together fMRI and anatomy, and Lupino was very important in this study. And so we know now that there are 2 streams coming from temporal lobe. One is coming from upper part of STS, the other from the lower part of STS. And probably this part, which is related also to semantic of the object, is useful, because when you grasp something, you must know something about the object. It's not that you are completely blind. However, that's the point that should be studied more. The important thing that we have 2 pathways, one going to area AIP, which traditionally was considered only for object, but there are a lot of mirror neurons, the area PFG, and then it goes to this ensemble. I don't want to go to detail, but area 5 has been now distinguished in different sections. Well, that's monkey, but we decided to move also to humans, and practically a couple of years after discovering mirror neurons, we went to Milano, where was a pet center, and we did the first experiment with the pet center. I must say it was one of the most, I was very emotionally involved in doing this experiment with pet, because it was really dramatic. It was the doctor running with radioactive substance, and we were looking what was going on in the brain. And after a while, you see that motor areas was active. Anyway, it was not like now that fMRI, you put just a patient and you do what you want. At that time, it was really with PET, rather emotional experiment. However, I will not show you those data, but something done recently, well, 5 years ago about, by Carl Zilles and his group in Munich. As you know, Carl is an excellent anatomist, and he made this meta-analysis showing in humans that you have an activation in the region which corresponds to STS, which is this one. It's posterior part of medial temporal gyrus. Then you have activation in the parietal lobe, and you have activation in the motor cortex and premotor areas, including in the human often is also the dorsal premotor cortex. It's interesting that from evolutionary point of view, it's very similar to the monkey area. They are maybe more.
This micro is not working.
It's not working.
Battery is run out. Use this.
Better now.
Está apagado. Dale al botón.
Okay, you can see here this meta-analysis made in Jülich, in which you see that it's very similar to the area which are activated in the monkey. And here is an experiment done many years ago in collaboration with Jülich, in which you see that there is also a somatotopy. So if you see a mouth action, you have an activation more ventral. For hand, it's more dorsal. And for food, it's going even upper in the superior parietal lobe. Okay, now I mentioned it before that in monkey, what counts is the goal. So we did an experiment using a robot arm, which has given us by Dario in Pisa. And here you see the activation when the head is grasping. Here is the activation, this one. One is the object is grasped by robotic arm. Remember that kinematics is different, and of course the shape is different. So there are many variables, yet the activation seems to be almost similar. If you do the subtraction, you find that in the parietal lobe, and precisely here, there is an activation which is specific for tools. This area, it's important for neurologists because lesion of this part or of the connection from this part to the motor cortex produce what is called ideomotor apraxia. So you are unable to use tools. There are many other symptoms, but important that you are unable to use tools. So we have there probably a model how to use tool, an engram, a motor engram how to use tool. So we presented to the monkey, to the human, to the students, other type of instrument, and always on the left side of the brain in the parietal lobe we have this activation. That's important because it indicates that even for something, I will come back to that later, that even for something which is obviously evolutionarily very late, we have already a model which allows us. One has been mentioned before, Tolman and his idea of model. I think I'm very close to this idea that we have model of things that then we are using for understanding what we are doing. However, one should be very careful. I have some friends which are too good and said everything can be understood by mirror neurons, which is not true. So we did an experiment that you can see here. We presented to students lying in the scanner action done by a person, by the monkey, and by the dog. The person was biting, the dog was biting, and the monkey as well. Everybody was doing the same action that all three species are able to do. Second part of the experiment, we showed this guy reading a newspaper, and you see only the lips moving, so no sound. The monkey was doing lip smacking. That's an affiliative gesture that the monkey perform when they don't know if you are good or you are an enemy. It's kind of a, stay quiet, I am good. The monkey makes, it's called lip smacking, ethologist knows very well this. And the dog was barking, again, no sound. Now, the result. Here is biting. If you look at the left hemisphere, which is the most important for this action, virtually, there is no difference. If I do a subtraction, it's zero. So when I look to a dog biting, my mirror system is active exactly as if I am doing the same action. So that's maybe on the basis why the people tend to anthropologize the animals and so on. But for some action, really, we are the same. Of course, it will be a spider there. Even with rat, there is much less sympathy towards rodents than towards the monkey. However, the interesting result was here. If you look at the man lip reading, there is activation of Broca's area, there is activation here of the temporal lobe. I mean, there is activation of typical oral communication areas. For monkey, there is very little here, and for dog was nothing, was the visual area. So barking, we don't understand using mirror system. We understand using inferential thinking. So like an apple which is falling down, and we understand it's going on because there are gravity and so on. When the dog, but think, can you bark? Of course not. You can imitate barking, you can make some sound. There is an interesting, I am a friend of a French philosopher, and he wrote me, my son, I asked my son, would you like to be a dog? And he said, oh no, why? Because I am unable to move my tail. Which is really true, it's really a deep consideration. We cannot do something which other animals do. In this case, we have to use our, we should become logical and do inferential thinking. So we know that dog is barking for some reason and so on. But that's very important. So we have two ways to understand the things. One is from inside, using our model and put what we see on our model. The other is just reasoning. So there is no controversy. As a matter of fact, now I have spoken with Fritz and the other people in England. At the beginning, people say, oh, there is a controversy, you have to understand in one way or the other. No, there is a kind of progressive thing. There is Goldman, Alvin Goldman is a philosopher who wrote a beautiful book about different ways in which you can understand people mentalizing, which you can do mentalizing, which start from mirror neuron, finish with complex inferential thing. Anyway, that's the point. That's with my friend Corrado Sinigaglia, we call it understanding other from inside. Now, I used fMRI in the monkey and in the humans. And of course, I was very happy of that. I may say also that when I read this paper, which has been done by a Danish group, Roland I was really elated because you can see what happens in the brain without actually, when the monkey is not doing, or the human being is not doing anything. It seems to be a miracle almost. And I think for many years, for about 10 years, this miracle worked very well. It worked well when? It worked well because at the beginning the people was very careful and was thinking also about real physiology or real neuropsychology. Then I think the things go a bit astray and the people now attribute to certain areas certain function and so on. But it's not my point to criticize. What I want to say, that was something wrong, not wrong, but limitation inherent to the system, and it's time. When you see one of those pictures that you saw before, time is not there. You see something, it's the end of the process. You don't know what is first, what is the second, what happened. You know what, that's the areas which become active when you do a certain action or present stimuli of different kind. Well, recently, about 3-4 years ago, we have been contacted by neurosurgeon in Milano, which do what is called intracortical recording in humans. That's technique which has been developed essentially in France and Italy, started in France, in Paris, but now it's very active, the group in Milan at Niguarda. It's technique which is used for surgery in people with epilepsy, which do not respond to drugs. Unfortunately, about 20-25% of people who is epileptic cannot be cured just by giving classical drugs. Giorgio Russo is the surgeon, Ivana Sartori is the neurologist who did most of the work. Pietro Manzini was the engineer who, together with other people, made all the software possible, because the difficulty, they have plenty of data, I will show why, but how to put them together, how to match them, it's really a nightmare. Now, the technique. The technique is the following. The electrode they use is this one. Maybe you can see here, 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 here are the space, the leads from which you can record. Up to now, we recorded not single neurons in the case, but gamma, very high frequency gamma, which correspond to action potential. It's not the same precision, but it indicates there is a lot of activity going on in terms of action potential. It's much more difficult to interpret alpha or theta or the other, but in this case it's easy. So they put this electrode, and they typically put 16 electrodes. For example, in this case, the doubt was the lesion is the temporal and parietal part, so they put all these electrodes like that, and each of them So 16 electrodes. In each of them, there are 10 points of recording. It means that we have in one patient 160 points. Of course, some of them are not relevant for us, some are in the white matter, but anyway, they are here. In order to be sure not to do damage, they do before the angiography, which here is in blue, but this is angiography, and the electrode is in between these. So technically, it's a rather complicated experiment. Now, what happens is they do first this insertion of electrode, then the patient recovers, and when they know where is exactly the focus, the epileptic focus, they do the surgery. So, but during this week, in which they record EEG day and night, and they record video, because they want to know what kind of seizure the patient has, the patient is available. And practically everybody said, if you want to work with me anyway, it's okay, we can do the experiment, ask me what you want, and so on. So we have a patient which is bored, it's there waiting, and it's ready to collaborate with us. So before showing you an experiment, I will show something which they do routinely, but we elaborated. They routinely stimulate the nerve, and here is the stimulation of median nerve. So here, it's simply a reconstruction of, 90 patients, I think. It's reconstruction of 90 patients, but time is not here. That's what you can see if you have fMRI, almost the same, because fMRI has other limitations. But anyway, imagine that you have MRI that you see, that when you stimulate the median nerve, you have an activation of somatosensory cortex, very strong, and then you have other activation. Now I will show you in time what happens. If you look in time, after 20 milliseconds, we have essentially the activation of primary sensory cortex, S1. But after 40 milliseconds, the hotspot, it moved to motor cortex. After 60, here practically everything disappeared. It's the motor cortex which is active. And after 100, you have an activation in the region around the perisylvian gyrus and also in the insula. So what you see in fMRI, actually, it's a misleading picture because it goes in time. And if you look at the type of responses, this one is it's small, but very prolonged. Now I will show you the same thing in animation. You see now, with time, practically disappear what happened there, and we see the perisylvian region and the insula active. So in other words, that's a very simple experiment. But even when you stimulate a median nerve, so it's a stupid thing, you see how complicated is the pattern in the brain. It's not that just somatosensory cortex is active. It's active also the insula, it's active the perisylvian region, and so on. Well, then we did some experiment, which we started, so we published only 2 or 3 papers up to now, but some are in press or in elaboration. So here it's hand action, and you have the black dots, which indicate when the hand, maybe I can show you first this. So the activity in the brain is synchronized either to the appearance of a tool of the hand, here could be a hand instead of tool, then when the action starts, and then at the end of the action. So there are 3 synchronizations: presentation of the stimulus, movement, and so here is when the hand is presented. You see at the beginning are these black dots. Then once the movement starts, you see the activation not only of the ventral sim, but also of MMT, and then it goes up in the intraparietal sulcus, arrive to the premotor cortex. And if you put on the contact, and then, that we did not expect, you have an activation in S2, and you have an activation in premotor cortex. So you see really a film. When you see an action, it's not something static, but you see the progressive. And here is the similar thing with a tool. With the tool, there is a lot of dots here. Remember that that's only the dots which are active. We have more than, I don't know how, 100, maybe 1,000 dots, because some are not active because are in the other part. But look at this region. That's exactly the same region which also in fMRI we see active, PFT, and then you have the same thing. If you present the same in terms of spots, you see all this stuff, but what I want to show is another thing. Here, it's now, what we are doing now, almost finished, it's time. If you look at the time, you see first the activation of this area, including MT, then this one, but not the region of tool, which became active much later. So when I see somebody grasping, I see first the action. I understand he's grasping, and only later I understand that this model is related to the tool. Here are the...
Over 90 subjects, you said, 90.
90 subjects, yeah.
Okay.
It's very long work.
Yeah, exactly. And can you say something about the variability across your subjects?
Well, the variability is destroyed by the fact that we have many subjects. Because, of course, it should be variability. That's one of the problems, how to match. So we use this, I am sorry, I have not explained something, nobody interrupted me, but here are these flat maps that we use because of the problem just been raised. First, because the gray matter is mostly inside the sulci. So if you see the Brodmann area, you don't see most of the gray matter. If you open, you see the gray matter. And then it's much easier to match in this way using this, the different maps.
And then here you look at, let's say, the high gamma contribution to the power spectrum.
Only the gamma contribution, okay. Exactly. Only one gamma contribution is higher than the rest.
Okay, but that's
synchronizing either with the beginning or with the action or with the end.
So the whole range of gamma from, let's say,
50, 150.
Okay, 50, 150, okay.
Yeah.
And is there anything within that range that stands out, or is it really do you broadly cover that range?
I have not.
50 to 150 hertz?
Yeah, that should be. So we make a power then of this.
But you broadly cover that range across all your subjects?
The whole brain is covered. We examine the whole brain, but in some part there is nothing. For example, in prefrontal cortex, if you do that, you don't see unless...
Well, I was wondering whether it was, for instance, maybe closer to V1 MT, you might be in low gamma, or if you move to motor, you might move to high gamma. So I was wondering whether there was anything like that.
That we have not examined, actually. No, that has not been examined. No, some part, following his question, some part are poorly explored. For example, this part here, V1 is not explored because the surgeon does not put the electrode in the primary visual cortex. Some other parts are difficult for the morphology of the brain. So not all the parts. I have a picture in which shows which parts are more explored and which are less explored. Now let me move in the last half an hour something else. What you see here is the insula. Insula is probably most of you know. It's a complex structure but has been considered related to emotions. There is also a literature which is a bit crazy because attribute to insula some highest function that you can imagine, conscious and so on. But let's forget, let's be neuroscientists. So if we stimulate the insula, that has been done by two my PhD student, Fausto Caruana and Gezzini from Lebanon. They stimulate the insula and found there are different regions. What I am interested at the moment is to speak about this most anterior part. Here is a red region, a blue region, and this green. What is here we have not understood because it was strange movement of the monkey. It was very inconsistent, but it was clear what happens in the red region and in the blue region. Now I will show you what happens when we stimulate this region in the monkey And now there is no action potential. What you have to look is just to the monkey and what the monkey does. The red light, when appears, the monkey is stimulated. The part of the insula I show it is stimulated. Red light. Red light. You see, every time this point is stimulated, this region is stimulated, you have ingestive action. It moves the mouth, it moves the lips, and so on. Now I move just slightly ventrally in the region which is blue, here. Obviously, she's not happy. Food, she looks at it. Not good. Again, that's very precious food for the monkey. Out. She's looking strange. It seems so good. Here we don't stimulate, so to reinforce her for a moment. Red light. Well, it's disgust. Something happened that the monkey became disgusted. There is study of roles, for example, and other people who show that in this region there are parts of it which respond to taste, other to structure of the food and so on. So there is a dorsal part which is related to ingestion, positive, and the other to negative ingestion. I don't like it. It's disgusting. I must say that a meta-analysis, again, done by the people in Jülich, showed that the ventral part of the insula is related to the emotion, while the dorsal is more related to other activity like ingestion and so on. Okay, that was monkey. Now we did with Vittorio Gallese and Christian Kaiser an experiment in humans, which preceded, by the way, this one, but it doesn't matter. That was done in France, in Marseille, using odorants. Because for a physiologist, using odorants is very complicated unless you are not trained to do it. So we applied to normal students some odorant in the nose, which could be pleasant, unpleasant, or disgusted. Let's forget about pleasant, concentrate on disgusted. That was the first part of the experiment. Second part of the experiment, they observed a video in which people showed disgust or showed pleasure or neutral. Again, concentrate on the movie in which this actor showed disgust. Here it's very clear, that's typical. Darwin described it, disgusted, you must do something like that. Okay, now the result. These are the result when you administer natural stimuli. Odorants in the nose, and the rotten eggs was the best stimulus possible, and you see this nice activation here. We were not the first to do that. It has been done before by people at University College, so it was done before. So this is activation. But what was really interesting is this one. Here you see what happens what happens in the brain when you observe a face disgusted and you have a real disgust. You see this white thing. White thing are voxels which are activated in both cases. So they are activated when you see a person or you feel a person. In other words, that's the best demonstration that this system practically interprets yourself as ourself. You and me are the same thing, because this is the point which, when activated, I am disgusted. But if I see disgusted, it's not that I make a logical conclusion. I see your face, therefore, but it's exactly the same thing. And remember that if I stimulate that, I have disgust. Also in humans, there are few experiments, but in France, the group of Magyar stimulated again epileptic patient, the rostral insula, and they have disgust. So it's me which are disgusted, and when I see some other person is disgusted. So really I understand you from the inside, not doing any inferential logical. This has really very important consequences, because this indicates that the same true for another structure, which I have no time to speak today, which is the amygdala. For amygdala, if you stimulate amygdala, you have fear, and if you see fearful stimuli, you have the same activation. So it's a mechanism which is known here. I presented this one because we studied. The other has been done by other. But it's interesting that if you have this mechanism, it's not true that this mechanism remain inalterate all the life. For example, if you are a surgeon, if you operate, this mechanism is blocked because you can do the surgery. You don't become compassionate towards the person who you operate. A policeman, the special forces, they are trained to somehow to suppress this natural thing. So we have the possibility. There are also some experiments which are a bit complicated, in which you train to people to consider somebody good and another bad, and then you see what happens in the brain, and you see that when the guy is bad, especially male, become very nasty. Female are more generous, but male become very nasty. So you have the possibility to modify this mechanism. But now think, just for a moment, then I will come back to science. Think for a moment on the social consequences. If you have an ideology, like the best example is Nazi, what happened in Germany in the 30s? If you think about Germany in the 30s, it's one of the most cultured, advanced country in the world. It's Goethe, Beethoven, you can mention them, and many scientists. How it's possible that in few years they were able to persecute a group of people, especially Jews, for no reason except because they're Jews? If you think sociologically, there's a reason. The Germans lost the war, and somebody has to be a culprit. Somebody is responsible. So the propaganda of Goebbels say, we lost the war because the Jewish were there, they were traitors, and so on. In addition, there were this, the economical situation was poor, so the people was happy. And if you read the Eichmann process by Arendt, the philosopher, it's incredible how Eichmann was not a bad man. He simply said, I obeyed the order, and I was just organizing and thinking, I have friends. But if you lose the capacity to think that he is exactly as you, but he is some Untermensch, that's the term which was used. You are not real man, you are Untermensch. Why you cannot organize the camps? Why you cannot organize for the good of your country the transportation, like Eichmann essentially was doing transportation of Jews from one part to the other, and so on. So it's very dangerous if you lose your capacity to feel the other as yourself using your biological mechanism, and consider that the other people are not human beings, but something similar. But at this point, what's wrong to put some trees and to burn the trees if you need warmth? So it's extremely dangerous thing, because I think our biological mechanisms are not inflexible, but can be modified by ideology. So ideological battles should be very important, without exaggeration. But anyway, one has to think that there is something there important. I will finish with my mirror mechanism, and now we go back to the cingulate cortex. Cingulate cortex. Oh my goodness, it disappeared my slides, just a second. I have to find. I will find in a second, excuse me. Signor. Okay, here is the cingulate cortex, and it has been, cingulate is one of the places where very often there is epileptic focus. So it has been explored many times. Now you see these points in which they stimulated, the surgeon stimulate this point to see what happens. And the black points indicate that there are movements. Essentially, this part is movement. These are visual sensation. But the interesting in this part here, this colored part produced laughing. Now imagine, this is a patient which should be operated in a few days, of course, with already the electrodes in the brain. So you stimulate this part and it starts laughing. Now we show you a movie in which is a girl. The girl is invited to read, and she read some word in Italian, and then at a certain point...
Lavoro coltello, maestra, ferro, gatto, arancio, pulsante, auto, porchino, no, un attimo di...
Da ridere?
Sì.
And it's compulsive. She can, if you ask why, there are two types of patients. Some try to rationalize and say, oh, something happened that's funny in my mind. The others say, I don't know. I started laughing, I don't know why. So this region is one stimulated, produce laughing. So at this point, we presented movies in which a guy was either crying or laughing and neutral. And if you see gamma, it's extremely strong when it's laughing. When you see laughing, you have an increase of gamma activity. So the same center, which when it stimulates, produce laughing, when you see laughing, it's excited. So it's again the idea that you understand laughing because your center for laughing becomes excited. To finish, so what I think the general idea that we are following now is that we have to go somehow away from classical empiricism of physiology, of English empiricism, the idea that everything is coming from out. I think the important things are inside us, and then experience, so on, modified and so on. But we recognize the people, we recognize emotion. So the newborn recognizes the expression of the mother. It's nothing learned. She already has a mechanism for recognizing the expression. The work of Stern, for example, is beautiful in this sense. So this laughing is not learned, it's there. And so you, by the way, I think that's a trick which many comedicians use. They start laughing before telling you the story. And in some American soap opera, you have this laughing in the background, which for me is extremely disturbing, but it seems useful. Okay, thank you.
Thank you, Giacomo. Do we have a microphone for the questions? All right, we can go on nevertheless. Are there questions, Giacomo? All right, Lisette, let's start here in the front.
Yeah. Who is first, her?
Yeah. I will speak louder. So
Yeah,
that
but
can-
we are close now.
Okay, so that was amazing. So most of the data that you have presented are average data from, but I wonder about individual differences and whether you can relate individual variability with different capability of empathy or reproduce what the other is doing.
Yeah.
So,
Well, the most clear individual difference I know is between male and female as far as empathy is concerned. If you think that in London, for example, Tanya Zinger and all this group, when they study empathy, they use only girls because they are, the results are much better than with males. So that's, I think, it's very clear. For other things, I don't know. For disgust, we don't find any difference. Disgust is very common to all ages, all persons.
and if you examine some psychiatric diseases, which eventually can affect that capability.
Well, here was a surprise. Jean Dessetti made an experiment using a psychopath in the jail in Chicago. And he showed scenes which should be very terrible. Instead, they recognized the emotion perfectly. And if you think it's not so strange, because if you are a sadist, to recognize that somebody is suffering, it's positive. So you recognize, but instead of having the reaction that we have, poor guy suffering, we have to help him. But remember that when I talk about empathy, it has nothing to do with religious idea of compassion and so on that became now very popular with the Pope and so on. Empathy is just to understand what is happening. Then how you behave, it depends from many factors. Even if you like so much your grandmother, but you don't know how to do it, you put it in the hospital and she's there. For her, it will be much better to stay at home. But if she's a bit demented, it's dangerous, so you put away. So not because you are compassion, but the empathy can, so you can react in a different way. And your question is very interesting because people think that people who are in the jail, well, they don't understand emotion. They understand very well emotion. At least this type of people, I mean.
Which
sad.
means the control of that emotion. When
You mean the girls from Tanya Singer understand emotion?
All right, so it's often a great talk, by the way. Mirror neurons are often seen as important for understanding others, right? But one could say, well, the mirror neuron itself doesn't distinguish whether others do it or yourself. They respond to a common action. So could you comment on whether you find neurons that do respond?
No, that's not true because, of course, if I do an action or I observe an action, I have many other cues. The first, when I do an action, I have my prefrontal lobe or cingulate which say, do it. Second, I have proprioception which say that I am doing the action. When I observe you, I have the same mechanism about the action. But of course, I know that it's you doing that.
Yeah. So the question is...
For emotion, it's more tricky maybe, but for action, for cold action, it's very clear.
Right. But could you comment on the system that does respond to action, but does distinguish the individual himself or herself and others?
So I already told you that I think there are many elements. When I do an action, I have the will, let's say, in brackets. But anyway, I have my action, which is determined by my frontal lobe, by cingulate, maybe by hypothalamus if I am angry. So, well, don't forget the subcortical center. They are very important. When I observe you, it's not so. Even with disgust or pain, I think that is different because you have the stimulus. In the other case, you saw it. Yeah. So you have elements. I don't think there is a possibility to confound them in the normal people. I don't know about possible patients.
All right, Giovanni.
Yeah, so thank you for the great,
Yes.
Thank you for the great talk. So it seems to me like there's like an element missing, like in the action.
Sorry, I was moving.
Sorry. There is like an element missing in the action understanding story that is like coding for action outcome. So I think that like often, to understand the action or a goal of an action, we also have like to somehow predict what is going to be like the consequence of the action. It's going to be good or bad. Also, like, especially also to learn from actions of other people. So my question would be like, do you find any neural population that responds better to action outcome of other people rather than to the actions?
Yes.
Thank you.
We have two experiments. One we did a couple of years ago with Fogassi, in which we demonstrated in parietal lobe there are some neurons which fire when you do an action for a specific purpose. So I grasp to put them out, there's a discharge. I grasp to put a container, no discharge. Most of these neurons have also mirror properties. So if I see you grasping for eating, it discharges already when you grasp the object, and instead if you put in the container, it's not. So there is a mechanism in the parietal lobe, and probably also in frontal, but we have few data on frontal. In parietal lobe, which allow you have a chain. Thanks to the chain, you predict the next move.
But what about, like, what if the action fails, for example?
What is that?
When the action fails, if the action fails.
Well, that was one of the most disappointing experiments I did, because with a lady from Torino, who is professor of psychology, she prepared a beautiful film in which there were action, in which, for example, one was playing chess or making a mistake, the chess was falling down. So we think, what happens? Then happened nothing. I mean, there was mirror neurons and attention. So when you see wrong thing, the attention was dominating. Plus, you have some mirror neurons because you move the head. But we have not discovered anything. It was trivial.
So there's still hope for me.
There's still hope for you.
Okay, Neil.
So both of these talks were fascinating, and I wonder if you would draw some links, perhaps. So in the hippocampus, we see cells for a place, irrespective of how you know you're in the place. And in humans, you see these Jennifer Aniston cells, irrespective of how Jennifer Aniston concept is produced for you. So the idea of sort of concept cells seems very related to both talks, in the sense that it's the concept of putting something in your mouth, which these neurons are representing. It doesn't matter who is doing which mouth, how the action happens.
Mm-hmm.
Do you see a link there?
I am a bit embarrassed
No, don't look at him.
talk about.
No, Giacomo, don't look at him.
I don't look at him. No, actually, I don't know. Even if Moser was outside the room, I don't know.
Do you see the link?
But you know maybe something.
You have something in mind, I'm sure.
Well, it seems related that all of your experiments, going from the idea that the neurons should be just coding for the action of the owner of the effector, to firing for the same action, or indeed the same goal, irrespective of the type of action or who is making it, sounds very similar to the neurons which respond to the concept, not of an action in this case, but of a famous person, or being in a place, or being pointed in a particular direction. They're all interesting cells because they're in the middle of the brain, and they're not dependent on the specific nature of the stimuli that's driving them.
No, I see the point. Yeah. But I don't know how to approach it experimentally.
You don't want to make any link. You could say that all these cells are, mirror cells. Equally, you could say all these cells are concept.
Well, I know that in Trondheim there is an attempt to find mirror neurons in the hippocampus, and maybe the success is moving.
Yeah, but
Better take a bit of microphone.
Microphone.
There is, which Neil also probably knows about. Nahum Ulanovsky has recorded cells that respond to another animal's location, which are partly overlapping with the traditional place cells. I don't think that's published yet, but several groups in different places in the world have seen this, so I guess it is somewhat similar.
That's very similar, yeah. Does that answer your question? Come on.
The other half is whether it's you making the action or somebody else making the action, is just another way to get at the concept of the action. And so you can see it as like reading Jennifer Aniston or seeing a picture of her or
Anyway, you're bringing a concept to mind, and some neurons are firing. These neurons are representing this concept. Is that a common way of thinking about all of these cells?
Well, they're all conceptual in a way.
I don't know, the danger is to move towards something which is really conceptual. So I want to keep very close to motor system to eliminate the idea that everything is conceptual, everything is inferential and so on. So I'm a bit worried to accept your point. Although I remember many years ago, Pilishin, who was a cognitive psychologist, one of the most famous, said me, but what you discovered is just conceptual neuron. It's grasping, it's a concept, it's not motor. Well, but it's motor.
But it's not motor also.
You see, that's the point. That's the point.
All right, Tony.
Can you say something about the development of the mirror system? And I mean, is there any link that you can see to, say, the development of theory of mind? And also in children and adults who have autism, is there anything that you can see in the mirror system that might reflect that?
So your point, if there are children with some problem with this system?
Well, I mean, if there is some link between the development of the mirror system.
Again, I am very cautious here because I think that there is a link. I think what happens in autism is for genetic reasons, the motor system tend to develop late and not in a good way. If you have sufficient stimuli to overcome this difficulty, you may be cured. But if you don't give, you become autistic. But the mirror neurons are a secondary consequence. It's not like the idea of somebody who had broken mirror neurons there for autism. I don't think it starts with broken mirror neurons. It starts with some more general deficit in the motor system. And if you talk, especially with the people who rehabilitate children with autism, they will tell you that all of them have problem with subtle-think, other stop moving for a certain point. So there are a lot, which typically doctors and psychologists consider it minor because you have this big problem of social interaction. But now there is a strong move towards this idea because if you can do the diagnosis early, your probability to improve the condition of children increase. There is Solly Rogers in the States, in Davis, which showing some films that if you do the diagnosis very early, then you manage staying a lot with kids, playing a lot with kids, to improve very much. But if you do just nothing, or the mother tried to ignore, saying, oh, well, also my grandmother was a bit retarded, so it's a disaster. It's unfortunately, often, although the parents of these children they like their kids, but they try to minimize, saying, oh, yes, also my cousin was a bit retarded, also my grandmother, and so on. So they tend to minimize, because it's a defense.
I don't know if you have enough data to say anything about this, but I wonder to what extent, what is the overlap between the subset of neurons that are active during observation and those that are active during when the animal performs their action or an action itself.
So the action itself and the observation.
Yeah, is it a random subset or is it discrete cells so that you can actually always tell if the animal is watching another animal doing something or doing the same itself? Is it possible to say anything about that? What is discrete subsets of cells?
No. We know that most of mirror neurons are in layer 3 and therefore they should be parameter of layer 3. In layer 5, most of them are motor neurons. But exactly, and sometimes you make a mistake because, for example, we describe that most of them are on the convexity. And now with the new electrode, in which you can record many, many neurons, we found several of them also in the bank. So sometimes, some data which statistically are significant are wrong.
I have actually one remark and one question. First, I want to thank you very much for the lecture and for the work. and because this is an excellent demonstration how much research in non-human primates can provide unique discoveries in neuroscience. I want to stress out that because today we are sometimes facing society or organizations that are not understanding why we perform research in animals, and especially in non-human primates, and we are in front of talented and motivated people and students. So this is really an extraordinary demonstration of how much monkey research could lead to a unique discovery in the history of neuroscience.
Yes, thank you very much for that, because really, I don't know how in other countries, but in Italy it's very difficult now to work on monkeys. Fortunately, we are somehow a well-established lab, we are not attacked. But for a new group to establish monkey experiment, it's a nightmare. I will tell you a story. When I was very young, my professor asked for the monkey lab, just calling the veterinary chief of our city, saying, can I do experiment of monkey? He said, yes. And who will be responsible? You, because you are the doctor. I know about cows, about other animals, pigs. Now, in order to get that, you need at least 1 year of work, of paperwork, and it's not sure that you will receive. You have to demonstrate it's necessary, that is no pain for the animal. It's terrible. We are still living a kind of happy island because we have monkeys since 30 years, and nobody attacked us up to now. I hope not after this talk.
Okay, then the question. The question was, how can you see the mirror brain and mirror neurons perspective in terms of predictive coding theory? Because we had actually we had experiment that we published 2 years ago in macaque monkeys using fMRI and playing auditory sequences with the high complexity of organization of these sequences and deviancies. And we really, each time we activate F5 very nicely with the high order sequence violations. And we try to interpret that in terms of predictive coding. What is your opinion about that?
Well, prediction can be used in 2 ways. One, what I told you before, somebody asked me about if there is a way in which you could predict because the monkey is grasping one thing to the other. The other way, more scientific, more, if you want, mathematically oriented, is the idea of Friston. Friston and the group in London, including Kilner, who is really expert of mirror neurons, they claim that they are not so important for understanding things, but they make hypotheses a priori. So you have some hypothesis that you are going to bring something, and when you see it, there is matching between this a priori thing. So our idea that you must activate this neuron, he thinks that the neurons are already activated, and then they meet together somewhere, maybe in STS, the stimulus coming. It's a very interesting idea, but people working in modeling claims that they should lose a lot of time. This system is very clever in principle, but they are not sure that it will work in real life because it takes time. But it's very, I'm very fond of this group. They're working very well.
Hello, and thank you for such a fantastic talk. I hate to bring up the conceptual again, but...
That's becoming because it's easier when I can hear well. Yeah.
Okay, but I was wondering if you could give some type of thought on if you think the functionality of mirror neurons could be attributed to more indirect forms of empathy, such as the triadic relationship between an artist, the artwork, and the viewer.
I've not understood. I can hear very well.
So you're asking for like a reinterpretation of the mirror neuron idea towards another context.
Right, exactly.
Right?
Which is more, it's more of an indirect kind of form of...
When I interact one person with another.
Well, triadic, so she wants the artist.
Triadic she wants.
With, let's say, a piece of art, like the artist interacting with a piece of art and an audience.
And the viewer,
And the viewer. But there are many examples we can invent now, right? So this will scale badly.
But you're thinking about figurative art or the performance? Because figurative art, there are some people think that in a sense what the artist put in the painting, then it's recovered by the person looking at it. So even some people think like Friedberg, that even the movement are important. If you think of some art, so you can recognize the movement of Pollock or of Fontana. So the idea that you appreciate because you feel inside yourself the movement which has been done. But that's, I think, I don't know, that's really speculation, even worse than my political speculation.
Come on. But you got provoked, it's okay.
I have provoked. Yeah. No, I think everybody agrees. I think.
Yeah, right.
I think nobody is a fan of Goebbels.
Come on.
Hello, thanks for the talk. Just a question, do we know if this mirror system is more innate or more acquired over time in infants? And if it is acquired,
you know, that's...
do we have any idea of what could be the teaching signal, the invariant in both the observed and the executed action?
You know, the people like to fight very much. So we have people who think that mirror neurons are innate, and there are people who think they are completely learned. I think both of them are right. So some of them should be innate, but certainly some you learn. For example, innate, evidence for innate is the Meltzoff experiment, and the Meltzoff showed this tongue protrusion and has been replicated even in the monkey, so I think it's true, or those some people. And if you read the development books, it's a lot of communication between the mother and kids, which is immediate, almost immediately at the birth. On the other hand, there is no doubt that as soon as you learn the new skill, then you understand better. There is an experiment done in Amsterdam in which they presented to child which is still unable to walk a film of child moving, and there was no desynchronization of the rhythm. Instead, if you show to the same kids a small boy crawling, and he is able to crawl, there is a desynchronization. As soon as the boys start to walking, it's very good. And then there is a famous experiment made in London by Calvo, Merino, Haggard, and the other about the dancer. So the dancer, if you see, if I am a dancer, it is some body dancer, there is some activation by mirror neuron, but it's not so big. If I am a professional dancer, it's a huge activation, but only if I am a dancer of ballet, which you observe, because there are professional dancers of classic ballet and those of capoeira. Those of capoeira, if you can dance classical dance, you have the same reaction as myself. So no reaction. But if they see the other. So that's clear that learning some skill, you know how to do it.
How this learning could happen? What is the training signal? What is the invariant, for example? Is it, for example, an higher order invariant? Or, for example, for the infant acquiring, having the same response, for example, for observing someone watching?
Now we are talking about, for example, grasping. Because different type of grasping, the invariance is very big because they are completely different. Yeah. That's why I stress the goal of the action rather the kinematics or the dynamics of movement.
Okay, thanks.
So Giacomo, one thing that worries me a little bit, or worries me, is I find it an interesting challenge. You say, look, we overcame this traditional empiricist view on the brain, right? Because now you also show there's internal structuring, if you want.
Yeah.
So how action is generated and the actions of others are interpreted. Okay, great. But on the other hand, if we look at your data, I could also say, well, but on the other hand, you are possibly facing a challenge of this traditional faculty psychology view where distinct functions are really localized, right? Because you look at the insula, there we see more, let's say, a valence kind of dimension, very locally expressed. Cingulate would be, let's say, your laughing center, and then we have, let's say, a premotor, parietal kind of system for more action-related, right? So it becomes very localized. So now, how should I think about, let's say, the mirror brain to transcend this idea of very local functions?
No, I don't see there is a contradiction, because when I say the mirror brain is because I have this center, which have an internal model of laughing, let's say. When I see you laughing, this center became active. Mm-hmm. So about laughing, there is different type of laughing. Maybe there is a center of laughing. You know, sometimes laughing is because you feel superior. So this laughing is completely different when you are laughing because there is a joke. So maybe there is different center for laughing, but certainly one is there because when you stimulate, you laugh, and when you show a laughing person. But so I am not convinced there is only one center for laughing or one center, but I think there is centers, and not that everything is learned empirically, that we are tabula rasa.
Yeah, but isn't that annoying to you? Because now we have qualitatively different models that in order for me to deal with you now, I must again link all these, I have to bind all these models together, right? So is there then a meta model that does that, or is there only a linking between these sub-models?
No, one point which should be clear, that unfortunately people still make a confusion, are convinced that the mirror system is motor system in premotor cortex. And so if you have laughing, so it should go through the motor cortex to back to cingulate and so on. I think they are completely separate center. So the transformation of sensory information, motor information, it's made in parallel in many completely different centers.
Mm-hmm. But how do I bind them now together if I want to respond to you? I say, oh, Giacomo was laughing because I told him my latest dirty joke.
Yeah.
Right. So how do I then bind these models together and the predictions that they make, and that might also be contradictory. So how do you bring it then together? So you're saying you have like a divergence, there are different models that run in parallel that allow you to interpret what you do.
I think they work in parallel. So I think when I mentioned, somebody asked me about attention, that's all completely in parallel. So we see that when you move your hand and you play chess in normal way, you have the mirror activity. If you do that and then you move strangely, so you have mirror neuron at the beginning and then attentional system, which say, oh, something wrong.
But who does the arbitration now between these subsystems? That's my problem.
Now he's pushing me towards philosophical issue.
No, an open
Which
question.
I don't know.
No, but
the arbitration.
It's an open question. So
the arbitration to decide. Well,
yeah, is it going to be attention or a mirror system response, right? Where are you going to look? If you have to address this.
Well, if I have a model how to play chess and somebody is doing something crazy, my brain immediately understands it because it's against the model.
Mm-hmm. All right.
So about sport, there are many examples. For example, professional basket player can know if the ball will go in the basket. Instead, even a good player often doesn't know.
Sure.
That has been studied and it's rather convincing.
Mm-hmm. Okay. So we have another question in the back.
After this philosophical question, it's finished.
Please, give me something concrete.
Yes,
so
please.
Here's a much more practical question. I mean, we can also imagine carrying out movements, and I was wondering if you consider these as sort of different, certainly phenomenologically different, of carrying out an actual grasping movement, watching someone else carrying out a grasping movement, and then imagining carrying out a grasping movement. Have you, or would you imagine seeing different signatures, maybe in where the activity is, the kind of activity?
No, that has been done. By the way, that's a very good example why I think that when we say understanding from the inside, because if you have motor imagery, using the general term motor imagery, you imagine to play tennis, or if you play tennis, or if you observe somebody playing tennis, practically the same structure which are activated, except the motor cortex, because for playing you have to use it. But that's something which is well known by sport trainers, especially in athletics, in which they say, repeat mentally how you will jump. And that's the same like jumping. So, but anyway, coming back to science, it's exactly the same structure. It's activated when I think an action, when I do an action, I observe an action, except the motor cortex, because you don't perform the action. No, but that's a very important point. For me, it's very convincing that really the mirror system is somehow me, because when I think of the action, it's the same structure which is activated. So it's, but that's something we discussed with each other many years ago.
All right, so thank you very much, Giacomo, also for your stepping up. So then I again want to thank Edward and Giacomo and all of you for being here, for having a great session this morning, where we learned a lot. Anna, you have some announcements?